Well, hello everybody, and welcome to the ESDR Kitchen. For those of you who are not too familiar with our kitchen, so it is actually a series of live events that the ESDR has been organizing for the past few months in order to promote scientific communication during those challenging times for some of us. So we have within the kitchen 4 series of events, the recipe book where new techniques are being presented, or molecular cuisine where leaders in their field share their personal journey through scientific discovery, the freshly baked series where recent groundbreaking discoveries are discussed, and finally the sweet and sour series where 2 protagonists discussed a controversial topic, and this is actually the episode of today. So it is my great pleasure to introduce our 2 presenters of today, Professor Birgit Lane and Professor Pierre Coulombe. Professor Birgit Lane is currently an A-star honorary fellow at the Skin Research Institute of Singapore. She has worked for A-star Singapore from 2005 to 2021. She set up and led the Institute of Medical Biology, was its executive director from 2007 till 2018, and then she initiated the Skin Research Institute of Singapore, which she co-directed with Roy Chan from 2013 to 2016. She has served as chief scientist at this institute until 2019. Birgit Lane's early career was London-based with a BSc and a PhD degrees from the University of College London, and then postdocs at the Imperial College, UCL, and also Cold Spring Arbor in New York. She moved to the University of Dundee, Scotland in 1991, where she helped to establish the Wellcome Trust Biocenter, and she held there the Cox Chair of Anatomy and Cell Biology from 1992 to 2009. Her research is well known, I'm sure, to each and every one of us. Research is focused on epithelial biology. She has made seminal contributions with her work on monoclonal antibodies to keratins and on the identification of genetic causes of skin fragility disorders, especially epidermolysis bullosa. She has over 260 publications. She's a fellow of the Royal Society of Edinburgh, the Academy of Medical Sciences in the UK, and the Singapore National Academy of Science, and an honorary member of the ESDR. Dr. Pierre Coulombe is a native of Montreal, Quebec, Canada. He serves as the Carl Huber Professor and Chair of the Department of Cell and Developmental Biology at the University of Michigan Medical School in Ann Arbor. He is jointly appointed in the Department of Dermatology, and he's a member of the Comprehensive Cancer Center at the same institution. Pierre joined the University of Michigan in 2017 following a 25-year stay on faculty at the Johns Hopkins University, including 9 years as department chair. Pierre received a PhD degree in pharmacology from the University of Montreal and pursued postdoctoral training with Ellen Fuchs at the University of Chicago. He played a key role, which is also, I'm sure, very well known to all of us. He played a key role in defining the vital role of mechanical support fulfilled by keratin filaments in epithelial cells and tissues. Was also involved in the identification of the first intermediate filament-based disease and the discovery of several non-canonical roles for keratin proteins, and this will be actually the main topic of his talk today. And in addition to a focus on the pathophysiology of rare monogenic skin disorders, research in the Coulombe lab has been increasingly focused on the basic biology of cancer and on the significance of the newly found presence of keratin proteins inside the nucleus. Dr. Coulombe has been very active in the education and training of graduate students and postdoctoral fellows throughout his career, and in the last several years in the recruitment and mentoring of junior level faculty. I'd like just to mention that this episode is sponsored by Almiral and supported by BMS. I'd like also to remind you that you can forward your queries through the chat or the Q&A functions. Birgit and Pierre, I'd like to thank you very, very much for your willingness to contribute to this session and to help us, or trying to help us, understand the complexity of the roles of keratins in cell biology. Birgit, the first part of the debate is yours, please.
Thank you, Ellie. That's very nice. Thank you for inviting us to be here. It's very hard to understand the function of proteins often because mostly their function is not fully visible in tissue culture cells, but it really comes to its own in tissues. And of course, that's not so easy to do experiments on. So for keratins, we have a number of advantages. First of all, they are generally very stable proteins. They're easy to isolate, good to identify. We have had the benefit of monoclonal antibodies to distinguish them for many years. So really, I think the place we start to look at function is to see what nature tells us, to see what we know out here. So as a result of work of many labs around the world, particularly Klaus Weber's lab and his colleagues, and we know quite a lot about the evolutionary pattern of intermediate filaments. For instance, we know that the diversification has really paralleled the increase in mechanical load, both of cells and tissues. For instance, there's very little in earlier, more primitive organisms, and the diversification really becomes apparent when we start to go to vertebrates, and particularly land vertebrates. So we have in mammals today, we have simple epithelial keratins, fairly small number, barrier keratins and stratified epithelia, and structural keratins of extreme tissues, such as the ones that, oops, oh no, sorry. There we go. The structural keratins, just to remind you what structural keratins do, they make very unusual hard structures that cohabit with soft pliable mitotic cells. Then we have some examples of extreme structural keratin structures in hair and nail and claw and so on. And for some animals, obviously the point with this is that these are once you've got the body functions sorted out, these are the things that come on the outside, the decoration. These are the things that determine the secondary sexual characteristics of animals, and they are going to be under acute selection pressure. So it's not surprising that we have a lot of these hard keratins or structural keratins today in mammals. Another thing we see immediately is that selective expression is very tightly linked to tissue differentiation. We have keratins expressed in pairs, obligate pairs, type 1 and type 2, and for each of those pairs, they're linked very clearly to very specific tissue differentiation types. In other words, they're linked to tissue function. So that's very clear from the work of many, many labs around the world through the years. So we have these points here. Evolving diversity parallels mechanical load in the tissues and the organisms, and selective expression is very tightly linked to tissue function. So this does rather suggest that they are mechanical or structural in function. We also know that keratin expression is upregulated with mechanical stress, even on a fairly short timeline. And this goes all the way from calluses and thickened skin that you develop on your hands and feet according to particular activities, whether you're a rower or simply walking around, and all the way to things like the rather ephemeral buccal occlusal line that develops between where your teeth meet in your cheek lining. This is mucosal epithelia, and there's a line of keratin 1 and 10 expression along that line where the teeth come together. So wherever there's friction or tension or mechanical trauma, you're getting an effect, a feedback through the keratin expression. So if you look at these filaments and tissues, you see very different patterns. Just examples here are keratin 8 and 18 shown in simple epithelial cells. And now I'm not sure where you can see. Can you see my mouse yet? No? Okay.
Yes, we see it.
Okay, fine. So this is two simple epithelial cells growing tissue culture, of course. Delicate, very beautiful filament patterns there. And you can compare this with the keratinocyte cell on the extreme right here, expressing keratin 5 and 14, also in tissue culture in a monolayer, and massive amounts of filaments here, densely round the nucleus and periphery linked into the desmosomes. And this is a blastocyst shown here with the same keratins as this kidney epithelial line here. In tissues, you can see simple epithelial cells in secretory tissues, such as this is a piece of the mammary gland. We have simple epithelial keratins in the secretory cells themselves, but backed up very often by a line of keratin 5 and 14, which is the structural keratins that are coming from the barrier tissue. So this is a much tougher phenotype. And in the epidermis, of course, we've got keratin 5 and 14 in the basal layer, and a switch to a different keratin phenotype on top. In the epidermis, it's keratin 1 and 10, which comes on as soon as the cells leave the basal layer, or sometimes even before. And this is these cells are post-mitotic. They're getting increasingly tough as they approach the surface. And in the end, the cells get cornified, of course. So very different phenotypes according to the function of the tissue. No argument about that. So the other thing to remember is that the keratins have to work in conjunction with desmosomes. Really important key element here. If you go back to old textbooks, you can see this in the just the H&E low resolution pictures of cells in the epidermis. This is a sort of classic picture of spinous layer cells, just to try and show you why they're called spinous layer, because you can see the spikes all the way around the edge, where this has just been, he's been referred to mostly as a fixation artifact because the cells shrink away from these links with the desmosomes. But actually, even in tissue culture, you see this very clearly. The bundles of keratin filaments come out of the cell periphery, where they're linked to the desmosomes, which you can see by here with double staining with high resolution bands of these beautifully symmetrical cell-cell adhesion structures. And there is definitely tension here. You can see that. Filaments go around the nucleus and they can often thicken there, but at the periphery, they're holding on to their neighbors and they're developing, pulling between them. At the high resolution level, you can see the filaments looped into these desmosome plaques. And this is, I think, the basis on which this structural function of keratin is working through this link with the desmosomes. But of course, the strongest evidence for function, for intermediate filament function, as with any protein, comes from human diseases. And here we're particularly lucky because we have a very clear example of keratinopathies, diseases in which the keratins are mutated, and this leads to a very dramatic breakdown in tissue resilience. So the poster child of this is the one that Pierre and I both worked on the first mutations of, epidermolysis bullosa simplex. And this is a dominant disorder, one mutation in one gene, either K5 or K14, the two partner genes in the basal cells, causes fragility of the skin. And you see here these classic clustered blisters on this child here, caused by scratching or rubbing or quite mild physical trauma. In the at the histological level, you can see that the basal cells have split, and this is stained with keratin 14, so you see staining of the keratin on both sides, meaning it's an intracellular break, very classic and key important thing. And you get these protein aggregates in the cell, more of which later. And then if you, very rapidly after this, of course, all around the world, people started to look at skin fragility disorders or epithelial fragility disorders and found mutations in most of the other major keratins. And they all have, they're called accordingly, but the main point here is just that if you have a breakdown in very superficial, if you have a mutation in a superficially expressed keratin, such as keratin 2, the breakdown is only in those cells. Keratin 10 and 1 here, you see breakdown all the way, not the basal cells, but everything above. And pachyonychic congenita, of course, is caused by breakdown, by mutations in the stress keratins, 6 and 16 and 17. So there's a very tight link between the expression of the disease phenotype and the expression range of the proteins. And now most of the major keratins have a clearly defined link with a known human disease, even down to not so clearly defined risk of gastrointestinal disorders coming up with the simple epithelial keratins, much smaller number, however. But you can't see those, of course. You only see the late disastrous consequences. You don't see the early lesions. So we have to make cell lines to look at these, cell models to study the mechanisms of pathology. You can't be studying the patients usually in these rare diseases. Either immortalized patient lines or cells transfected with a mutation can give you a very good model. And these are excellent reporter cells for drug discovery, as we've been working on over the last few years, but I'm not talking about that today. You see aggregates as you do in the electron microscopy of the patients. You can see them in the cultured cells, whether it's keratin 14 or there's a pachyonychia model with keratin 16 mutation here. And then you can use these to test all the different ideas people have about why these cells break down and what's going on. So how do these mutations lead to fragile skin? Well, surprisingly, it turns out that these filaments can still assemble, and they can still resist mechanical stress, at least stretch. And the pictures here are first from a very nice piece of work from Harold Herman's lab, a lot of work actually. And the second one on the right is from an experiment, so a series of experiments with Doug Fudge, which was done using RABS cells. And you can see these cells can be stretched to enormous lengths, and then they collapse back again. And the cells and the filaments have not broken, they haven't fallen apart. These cells have managed to do this. The in vitro assembly of Harold's, the key thing here is that these are pure protein populations, so they're not piggybacking on another filament system in the cell. So it's not that simple. Moreover, David Russell, who was a student at the time, also did experiments on oscillating stretch, and these found that the keratins were disrupted, but only in the EBS cells, not in the wild type. And you can see here the breakup of the keratins and the breakup of the plaque proteins to pull them into the cell, and they associate with keratin fragments further in the cytoplasm. So this is also stretch, but it's oscillating stretch, which means that the cells relax in between the stretch. And this led us to think that the signal actually might be the loss of tension, not just the stretching. So there's a lot of evidence from all sorts of studies, our lab and many others, to suggest that EBS cells are under constitutive stress, whether it's ER stress due to a misfolded protein responses, increased activation of stress kinase pathways, faster response to wounding, resistance to apoptosis. All of these things suggest that the EBS cells are in a state of constitutive stress. So we wondered whether all stress in keratinocytes might actually activate a wound response, because many of these things are exactly what you see when you activate keratinocytes. So we sort of thinking about this kind of a model here where you have mutant protein, which leads to aggregate formation, probably because the assembly of the filaments is slightly slow, or maybe has messed up kinetics. And these aggregates are recognized as misfolded protein and lead to stress in the cell. And we think that stress in keratinocytes is going to be interpreted as a wound response, and I'm sure, I don't think Pierre would disagree with this. This leads to junction destabilization because you're remodeling the keratin, and the key to this is the interaction at the desmosome interface, I think. And then the keratin remodeling leads to accumulation of aggregates, and that leads to increased stress. So you've got this vicious cycle. But that's all very well, but you need to be able, if this is how the keratins may be working in a very structural way, but you do need to, this is a pathogenic situation, which maybe isn't one that you would aim to have, except for the remodeling for migration. But you also need to remodel keratins under other circumstances, so that the hypothesis is only valid if you know you have systems in there to take the whole system apart rapidly when you need to do that. So apart from the migration remodeling, another classic is where you need to separate daughter cells for mitosis. And that indeed we have been looking at recently, and Vivian Wu made some nice antibodies which recognize phosphoserine that localizes to the cleavage furrow in the cells. So we know that, just to remind you, these keratins break up into aggregates during mitosis. But as they start to form again, if you don't have this ability to phosphorylate this particular serine by actually aurora kinase in the cleavage furrow, then these cells will not separate properly, and then we get an increase in these mega cells, multinucleate cells. Now this actually only works with keratin 14 and not keratin 10, because the sequence context doesn't allow the misfolding of the head domain in keratin 10, which happens in K14, and therefore probably helps to keep the cells unpolymerized during the end stages of mitosis. So in support of structural loads, we have many features here. We have the evolutionary diversity with increasing mechanical load. We have also other things which have come from other labs, like the very nice demonstration from Thomas Margin's lab, and that the epithelial cells with no keratins or defective keratins are much softer than the ones with the normal keratins. We know that cells with mutant keratins have less stable connections with desmosomes. We know from Paul Janmay's work that the keratins and other intermediate filaments exhibit strain hardening, which is another very good way. If you're tensioning these cells, you can get a very tough network across the whole tissue and dissipate the stress rather than focus it on one cell. And then we do have rapid and specific mechanisms to dissolve this when necessary. So I think the signal transduction mechanisms may well turn out to be a loss of tensions, and the desmosomes are critical. But now Pierre's going to tell us a lot more about something very interesting. Okay. You stop sharing, if I can find my...
Thank you very much, Birgit. Great talk, and we'll move to our next speaker, Pierre.
Good morning. At least it's the morning here in the United States. I'm so thrilled to be here. Thank you, Birgit, for this account of intermediate filament biology and intermediate filament function. I will give a slightly different perspective that's complementary to what Birgit shared with us. So my view of intermediate filament function, including keratins, is that there's two general flavors of these functions. One of them is about mechanical support and cytoarchitecture, and those are the functions that you expect on the part of cytoskeletal proteins. And the relevance of these is that when mutated, it causes skin disease that have a fragility component to it. But there's another facet to the function of intermediate filaments, which we've been discovering for the last 20 years or so and continuing, and that has to do with roles that are unconventional or non-canonical, and that often involves participation in signaling pathway. And in that setting, intermediate filaments can be disease modifiers. So to sort of sum up the classical function of keratins, these keratin proteins are very, very abundant. They love to polymerize. They're extremely efficient at it. They achieve organizations into intracellular networks that put this full potential into action. And then from this comes the structural support function. That function is what is at play with K5, K14 in the basal layer, K1, K10 in early differentiating keratinocytes, and in K2, a late stage differentiation specific keratins in the epidermis with the disease counterparts that we've already heard about from Birgit. In order to illustrate the other type of functions for keratin filaments, I would like to turn to K6, K16, and in particular K17 for today, which are constitutively expressed in ectoderm-derived epithelial appendages and are well known for their inducible expression after acute stress such as wounding, and in the context of chronic disease, including psoriasis and various types of carcinomas. K17 in particular has a long-standing association with carcinomas, and with the advent of molecular genetics studies, we are increasingly aware that high K17 expression correlates with a poor prognosis. So several years ago, my lab set out to ask what would be the relevance of K17 expression during the carcinogenesis using a transgenic mouse approach. And by the way, transgenic mouse models have been really important for function discovery and disease modeling when it comes to human disorders. So just summarizing about 15 years of work that took place in my own lab, if you examine the course of tumorigenesis in mouse models, what you find is that irrespective of the modality of carcinogenesis, we've tested GLI, which is relevant to basal carcinoma. We've tested HPV16. We've tested two-step chemical carcinogenesis in mice. In all these settings, the absence of K17 results in a delay in the onset of tumorigenesis and a change in the character of the tumors. In the setting of those studies, we have come to appreciate the fact that K17, which is at the time known as the cytoskeletal proteins residing in the cytoplasm, participates in the regulation of gene expression with a subset of genes having to do with inflammation and immune response from the standpoint of keratinocytes being consistently regulated in a fashion that's K17 dependent. We've implicated two interactions in those events. One involves the ribonucleoprotein HRNPK, another one involves the nuclear transcription regulator AIR. In the setting of those studies, we have queried and discovered that K17 occurs in the nucleus. It had been missed by all of us, including our lab, for years. You have to apply tricks because of the abundancy of the keratin filaments in the cytoplasm that sort of interferes with the ability to see it in the nucleus, but one can apply several biochemical tricks and find that K17, a small fraction of the K17 pool, does occur in the nucleus where it participates in a variety of nuclear processes. I would like to now share a recent study that came out of my lab where we have interrogated whether K17 might participate in DNA damage and repair processes, and that might be the basis for its association with the risk for carcinogenesis. And what we have found is that, as is the case following many types of stresses, which injury, UV exposure, DNA damage stress results in an induction of K17, including in the nuclear pool of K17. And if you assess the ability of human and mouse keratinocytes in culture to respond to various modalities of DNA damaging agents, the response is grossly abnormal. In fact, the response is attenuated. We found that biochemically, K17 exhibits a bias for the repair of double-stranded DNA breaks. And in the setting of the studies, we've discovered that K17 interacts with several proteins with known role in DNA damage and repair, gamma H2AX, 53BP, DNAPK, and so forth. We've also found that the absence of K17, if K17 is not there when a keratinocyte, whether mouse or human, experiences DNA damage, the cell will die. So K17 again manifests a pro-survival role. It manifests a rapid repair response, which is imperfect and sets the stage for a higher risk of developing cancer down the road. So a couple of examples. So this is a set of studies in culture with human keratinocytes where we have a CRISPR-Cas9 knockout in K17. We come in with the DNA damaging agent, and we're looking at gamma H2AX, which lights up the nuclei of the parental cells, which express K17. However, in the K17 deficiency case, we have no signal at this particular time. And that availability of a human cell model with a knockout allows for rescue assays. If you put back K17 in these cells, you have beautiful cytoplasmic networks containing K17, but you do recover the sensitivity to a DNA damage agent. But if you do the rescue assay with K14, it does not work. And if most particularly if you do it with a version of K17 that cannot go to the nucleus, it won't work. Likewise, we've been able to use proximity ligation assays to co-localize key effectors of DNA damage and repair in keratinocytes. Here I'm showing you at the top here data with gamma H2AX, which is a universal marker of DNA damage, and 53BP, which is particularly important for double-stranded DNA break repair. After DNA damage in K17 expressing cells, you have a lot of dots in the nuclei of the cells, but this does not occur in the absence of K17. And if you look at the interaction between K17 and DNA damage and repair effectors, like in this case here, 53BP, you see that K17 co-localizes with 53BP at sites of DNA damage in the nuclei of K17 expressing cells, but not in K17 null cells. And as I said, the price that one pays for not having K17 at the time of experiencing stress, including DNA damage, is that the cells is going to die. So K17 promotes the survival in the face of multiple types of stresses, and in this case, DNA damage, and if it's not around, the cells die. However, survival in this particular paradigm comes at a price. K17 promotes imperfect DNA repair, and it sets up the stage for mutagenesis that may lead to carcinogenesis. And we think that this accounts for the fact that high-level K17 expression correlates with a poor prognosis for several types of human carcinomas. So then coming towards the end, comparing what we've heard between Birgit and I, K14 is, can fulfill a structural support function, classical for cytoskeletal protein. This is filament dependent. It occurs in the cytoplasm. It involves a large fraction of the cellular pool of K14, which is huge to begin with. It is on all the time. That function is ever played, and it involves key partners such as K5, as well as adhesion proteins, as we heard from Birgit. If you mutate K5 or K14, you have fragility, and in this particular case, it's epidermolysis bullosa simplex, and the relationship to stress is a preventive relationship. With K17, we have evidence its presence of the nucleus, the fact that it's involved in DNA damage and repair. It is not 10 nanometer filament dependent. It is in the nucleus. It's a very small fraction of the pool. It's highly specified through post-translational modification. It's a transient phenomenon. It involves interactions with specialized effectors in the nucleus. It's relevant to cancer, and it does not involve mutations in K17. It involves its overexpression and its misregulation. And K17 response to DNA damage is an adaptive response to favor survival. So I would like to thank the organizers for their invitation. I would like to thank Professor Lane for graciously accepting to do the sweet and sour thing with me. And I would like to thank the members of my lab, past and present.
Thank you very, very much, Pierre and Birgit, for these two wonderful talks. And I think that they really nicely illustrate the fact that there is probably not one answer to the question that you were asked originally to address, which is what is more important, the structural or the non-structural roles of keratins. They apparently are connected. There is one short question regarding, because we are a little bit beyond time, but one question is how, so we understand how cancer is related to the non-structural roles of at least one keratin, K17, as you beautifully showed, Pierre. But are there any data connecting the structural functions of keratins to carcinogenesis? Pierre, maybe you may start with.
Not to my knowledge, but I think that everything that Birgit has shared with us is relevant to cancer. I think that we have implicated K14 in a role of mechanosensing through hipposignaling. Hipposignaling is a pathway that's very important to regulate the balance between proliferation and differentiation. It's heavily activated in cancer. So I think that the role of keratins in mechanosensing and mechanotransduction is likely to be very, very relevant to cancer.
And I think there's quite a bit of evidence that if you reduce the amount of keratin in the cell, it actually is better at getting through small spaces, that cell. So it would favor invasion in a cancer situation. I guess that's Thomas's collaborators, wider group. But I think definitely and also things like, yeah. I mean, if the cells have a good keratin 5 and 14 network, they're sticking together as a sheet very firmly. If you look at mammary cancers, there's also evidence that the cells that migrate from tissue, from histology, you can see it. You can see these Indian file cells, and they let go of their neighbors, and they can migrate in along the tracks, leading to around the blood vessels if they don't have a lot of keratin. So I think there's quite a lot of circumstantial evidence that too much keratin helps protect you from cancer invasion. Great.
So I would like once again to very much thank you both, Birgit and Pierre, for the most inspiring debate. And I'd like to invite everybody to our next ESDR Kitchen episode, which is due July 14th. This will be presented by Seiko Nakajima, and we'll deal with mouse models of atopic dermatitis, so please don't miss it. Please do not forget as well the deadline for early bird registration for our next virtual 15th annual ESDR meeting, which is due 22 to 25 of next September. You may also send us your happy birthday message video, and I might at the end get part of a movie that we are preparing for this very important celebration. So thank you all very much for having been with us wherever you may be. And once again, thank you very, very much, Birgit and Pierre, and see you very soon. Goodbye.
Thank you. Bye-bye.